After the very first International Summit on Human Gene Editing in December 2015, a declaration was released. The organizers were unanimous in agreeing that the development of genetically modified kids was irresponsible unless we knew for sure it was safe. Well, a great deal of good that did. As MIT Technology Evaluation exposed in November last year, a Chinese researcher He Jiankui modified embryos to develop 2 genetically modified infants. Other groups are now actively seeking to use the same technology to enhance human beings. This has actually triggered a few of the greatest names in gene editing, a few of whom signed the 2015 declaration to call for a global moratorium on all human germline editing sperm or egg cells, so that the modifications are hereditary. In an open letter in Nature today, significant players in CRISPR's advancement, consisting of Emmanuelle Charpentier, Eric Lander, and Feng Zhang, have actually been signed up with by associates from 7 various nations to call for an overall restriction on human germline editing up until a global structure has actually been concurred on how it must be dealt with. They recommend 5 years might be appropriate. The United States National Institutes of Health has actually likewise backed the call. The signatories hope a voluntary global moratorium will stop the next He Jiankui from unexpectedly springing another undesirable surprise. The group states that this moratorium duration will enable time to go over the technical, scientific, medical, societal, ethical, and moral issues that must be considering prior to the method can be utilized. Nations that choose to go on and enable germline editing ought to do so just after informing the general public of the strategy, participating in worldwide assessment about the wisdom of doing so, and ensuring that there is a broad societal consensus in the nation for beginning on that course, they state. The world might conclude that the clinical use of germline editing is a line that should not be crossed for any purpose whatsoever, the group states. Alternatively, some societies might support genetic correction for couples with no other way to have biologically related children, but draw a line at all forms of genetic enhancement. Or, societies could one day endorse limited or widespread use of enhancement. The letter's signatories recommend that germline research study ought to be permitted, so long as there is no objective to implant embryos and produce kids. Utilizing CRISPR to deal with illness in non-reproductive somatic cells, where the modifications would not be heritable, ought to likewise be great so long as any grown-ups getting involved have actually provided they notified permission. Hereditary improvement must not be permitted at this time, and no scientific application performed unless its long-term biological consequences are sufficiently understood, both for individuals and for the human species, they said. We still don't understand what most of our genes do, so the dangers of unexpected repercussions or so-called off-target results, great or bad, are big. The loss of the CCR5 gene that he was targeting to safeguard kids from HIV, for example, has actually been linked in increased issues and death from some viral infections. Modifications in a genome may have unexpected results in future generations also. Attempting to reshape the species on the basis of our current state of knowledge would be hubris, the letter states. The proposed moratorium and global structure are just voluntary, and not likely to stop researchers. However, the signatories feel a straight-out restriction and guideline would be too rigid. Rather, they hope their proposition will place major speed bumps in front of the most adventurous plans to re-engineer the human species. Undoubtedly, chastened by the He experience, China's health ministry is currently producing some speed bumps of its own. Recently it prepared standards that will require the country's researchers to look for approval from authorities prior to performing any dangerous treatments such as germline editing.